Hello everyone, and today in this virtual lecture I will be talking about the origin of eukaryotes. And here is just a picture of what a basic eukaryote looks like. You got your nucleus, your smooth endoplasmic reticulum, your rough ER, your ribosomes, Golgi apparatus, etc., etc., mitochondria. And just a quick review of what a prokaryote and a eukaryote is. A prokaryote is a simple organism and it lacks a nucleus or any membrane enclosed organelles. Okay? It does contain genetic material but not an membrane enclosed organelle. Eukaryotes, on the other hand, are more complex and they contain a nucleus and membrane enclosed organelles. So opposites basically. Now the origin of eukaryotes. Eukaryotes arose during the Proterozoic eon, which began about 2.5 billion years ago and ended around 543 million years ago. Okay. It's not fully understood how these cells first originated, how the first cell originated, but scientists have tried to determine this by examining DNA sequences. And studies show that the genomes of the nucleus, mitochondria, and chloroplast, which are the organelles that contain genetic material, are derived from cells which were once separate but then came together in some sort of event. Now, as I just said, eukaryotes contain DNA and nucleus and mitochondria, and plants, which are eukaryotes as well, contain another genetic containing organelle, which is a chloroplast. Now for my first question, genes involved in transcription and translation are derived from A. Ancient bacteria, B. Primitive eukaryotes, C. Ancient archaea, or D. Cyanobacteria. Now, this is just gonna, help you get warmed up for what's gonna come. It's a pretty straightforward question. If you have to, check the ebook, and you can pause this so that way it'll give you more time. Now, the correct answer is C. Ancient archaea. Okay, so the origin of our nuclear genome, it's derived from ancient bacteria and archaea, and it's believed that the ancient bacteria and archaea provide a lot of the material for the current nuclear genome of eukaryotes. So starting with bacteria, it's believed they provide the genes that encoded for lipid biosynthesis and metabolism, while archaea, on the other hand, encoded for genes that were related to transcription and translation. And so now some possibilities to describe the relationship between bacteria and archaea can either be either symbiosis or endosymbiosis. Okay, so symbiosis can be described as when two different species live in direct contact with each other. And the scientific hypothesis as to how this could have brought about the nuclear genome, well, an ancient bacterium and a archaeon could have formed a symbiotic relationship, they would have formed a close association with one another. And then a fusion event would have occurred and would have combined their genetic material, and which would then this would then allow for the retention of genes that were involved in activities such as translation, transcription, metabolism, and lipid biosynthesis. Okay. Endosymbiosis this can be described as a relationship in which a smaller organism, an symbiont, lives within a larger organism, a host cell. Now, the scientific hypothesis for this was possibly an ancient archaeon engulfed a small bacterium into its cytoplasm. Now, this possibly could have occurred via endocytosis, which is when the cell membrane invaginates or encloses upon it, or inverts upon itself to take in an outside substance into the cell. Now, once inside the cell, it would be maintained within the cytoplasm of the host cell, and eventually the genes from the endosymbiont would be transferred to the host archaeon, to the host cell, which will result in the formation of a nuclear genome. Now, mind you that the host and the endosymbiont are dependent on one another for nutrients, same as in a symbiotic relationship. Now, here is a pretty good diagram of symbiosis and endosymbiosis. I'm not sure if you can clearly see what's going on, so I'll just break it down. It's just going over what we just discussed. For symbiosis, first the close association is formed, and these two cells become fused. The genes that are desired are then there's the retention of the genes that are desired, and then later on we'll get into the discussion of how possibly another bacterium would cause the formation of a chloroplast. And then what we just talked about, endosymbiosis, a larger cell engulfs a smaller cell. The genetic material from the smaller cell is transferred into the larger cell, and allowing for the larger cell to take on new properties. Okay, and then again, a bacterium could later on come in and create a chloroplast. So these are the two main hypotheses as to how the nuclear genome came about. Now, the origin of mitochondrial and chloroplast genomes. In 1905, a Russian botanist suggested that these had an endosymbiotic origin, and not until the discovery that chloroplasts and mitochondria actually contain genetic material was this idea actually considered. And so it's believed that mitochondria are likely derived from a bacterial species similar to alpha proteobacteria. And this is a type of bacteria that utilizes oxidative phosphorylation to make ATP. So you can see why that would be a useful ability. Okay, so two possible hypotheses. First, an endosymbiotic event involving an alpha proteobacteria resulted in the first eukaryotic cell, and then the first mitochondrion was just a remnant. So the event happened, and then a mitochondria just happened to show up, if you want to put it in that term. Or a symbiotic or endosymbiotic event. So either one produced the first eukaryote, and then a subsequent endosymbiosis produced the first mitochondria. So basically what you're going to have here for the second one is either symbiosis or endosymbiosis occurs, and then the resulting eukaryote then would then engulf a smaller bacterium of some sort, or smaller organism of some sort, that would then produce the first mitochondria. Okay, so DNA sequencing of the mitochondrial and the chloroplast and prokaryotic genomes has been used to determine if these hypotheses are correct or not. and data from this sequencing has indicated that chloroplast